Constituent Factors Observed in Viral Diseases, Part 2 of 5. Pause text and illustrations for detailed study. Refer to the previously posted Constituent Factors Observed in Viral Diseases, Part 1. Cell membrane illustration. A virion is a fully assembled complete extracellular viral particle that infects a living host cell. A virion can be referred to as a parental virus since it gives rise to multiple progeny that tend to be genetically and structurally the same as the parent virus. It consists of a nucleoid constituting a nucleic acid core surrounded by a single or double protein shelled capsid that protects the nucleic acid of a structurally complete virus. Lipoproteins are combinations of proteins and lipids in soluble complexes. Lipids travel in the bloodstream in this complex. This nucleocapsid comprises the nucleic acid nucleoproteins, associated basic proteins, lipoproteins, protein enzymes that catalyze cellular reactions, and the entire specifically functional viral genome. Virions protect viral nucleic acid from digestion by nucleases. Virions have surface recognition sites that recognize and adsorb them to receptors on the host cell surface. Certain viruses have specialized proteins enabling virion penetration through the host cell surface membrane, or in some instances, the injection of infectious nucleic acid into the host cell interior. Human Immunodeficiency Virus HIV Virion illustration. Integrase is an enzyme of retroviruses that facilitates the insertion of its viral DNA into the DNA of the host cell, causing infection. Retroviruses are RNA viruses that, after infecting a cell, convert their RNA, catalyzed by the enzyme reverse transcriptase, into a DNA copy of their genes. After infecting the host cell, this viral DNA becomes incorporated into its DNA, allowing for the replication of the retrovirus. In the process of transcription, a single-stranded RNA molecule genetically complementary to one strand of a double-stranded DNA molecule is enzymatically synthesized. Thus, DNA is copied into RNA during gene expression. Reverse transcription is the creation of a double-stranded DNA molecule from a single-stranded RNA template catalyzed by the enzyme reverse transcriptase in a process that is the reverse of transcription. Gene expression drives virus formation, host cell infection, as well as further viral proliferation and spread. Human Immunodeficiency Virus Replication Cycle illustration. Illustrations of three basic viral shapes. 1. Helical virus illustration. A helical virus consists of a rod-shaped hollow capsid surrounding nucleic acid, seen in this tobacco mosaic virus illustration. 2. Polyhedral virus illustration. A polyhedral virus possesses a many-sided shell capsid surrounding nucleic acid, often in the form of an icosahedron. An icosahedral virus consists of identical, symmetrical, equilateral, triangular subunits arranged in a symmetrical manner, seen in this adenovirus illustration. 3. Complex virus illustration. Complex viruses are of a combined icosahedral and helical shape. This respective head and tail morphology is unique to viruses that solely infect bacteria. The tail of this bacteriophage punctures the host cell wall, comprising a channel that inserts its viral DNA. The bacteriophage is of a prolate icosahedral configuration, having an elongation on one axis, as well as its characteristic helical head shape. A bacteriophage is a parasitical virus that infects a bacterium and uses its biochemical machinery to reproduce other infectious viruses capable of infecting other host cells and causing disease. Illustration of coronavirus disease 2019 virion structure. Lipids are water-soluble fatty or fat-like substances contained within the structure of cells serving as a fuel source that are supportive of other biological substances. Phosphorus is an abundant chemical element in all tissues that is involved in nearly all metabolic processes. Phospholipids are phosphorus-containing lipids comprising the principal form of lipid in all cell membranes. Some viruses possess a phospholipid envelope surrounding the protein capsid that is derived from the infected cell membrane of the host. Viral genomes encode four major envelope proteins essential to structurally complete viral particle formation and replication. Small integral envelope membrane proteins participate in the assembly, envelope formation, pathogenesis, and budding of progeny virus from infected host cells. Spike projections are generally viral encoded dimeric or trimeric glycoproteins that are inserted into the lipid envelope involved in receptor recognition. Dimers and trimers, respectively, are polymers consisting of a chemical compound or molecule consisting of two or three simpler identical individual molecules. These spikes are also referred to as peplomers that protrude from the envelope of certain viruses. The spike S proteins mediate virus attachment to the host cell surface receptors and subsequent fusion between viral and host cell membranes, facilitating entry of the virus into the host. Spike protein expression at the cell membrane is capable of mediating cell-to-cell fusion between neighboring infected and uninfected cells. The membrane M protein determines the configuration of the viral envelope, interacting with the other structural proteins. The spike S, nucleocapsid N, membrane M, and envelope E proteins are functional elements in the life cycle of the beta coronavirus SARS-CoV-2, causative of COVID-19 infection. SARS-CoV-2 virus surface spikes illustration. Influenza virus surface spikes illustration. Human immunodeficiency virus surface spikes illustration. The nucleocapsid N protein binds to the viral RNA genome comprising the nucleocapsid, as well as functioning in the virus replication cycle and the host cell response to viral infection. An external lipid membrane bilayer surrounds the envelope surrounding the capsid of some viruses. Glycoproteins are bonded protein carbohydrate conjugates by definition, functioning in unison. The lipid bilayer is derived from the modified host cell membrane and is studded with an outer layer of viral envelope glycoproteins. The envelope contains specific viral proteins that function in the binding of the virus to host cells. The protein coat protects the nucleic acid from environmental damage and allows virion attachment to the host cell membrane. There are host cell surface receptors that are specifically recognized by viruses, upon which viral binding occurs. Cells lacking these receptors are not infected. Upon entry into the body, the specific viral particles attach to the particular host cell receptors. Transfection refers to the introduction of foreign nucleic acid into a cell. The viral genes are transfected into the host cells via the receptors. Sensitivity to infection occurs when there is transfection within the gene coding for the receptor. The nucleic acid contained in the capsid is released in the infected cell, and virion components are synthesized, and gene transcription and translation result in replication and assembly of progeny virions capable of leaving the cell and infecting the neighboring host cells and tissues. Illustration of viral tegument. A viral tegument layer present in the cytoplasm is a structural viral encoded protein cluster in the space enclosed by the lipid envelope outside of the protein capsid that releases viral DNA into the nucleus. Tegument functions in viral DNA delivery to and replication in the nucleus, facilitating viral entry, gene expression, virion assembly, immune system evasion, as well as virion egress and spread, infecting other host cells in the presence of the immunoregulatory antiviral glycoprotein interferon. This host cell signaling protein cytokine exerts virus nonspecific but host specific antiviral activity in the presence of a virus. This is particular to the herpes virus. Interferon can also suppress the blood supply and cell proliferation of tumors, as well as activate macrophages and natural killer cells in the immune system response to cancer. Interferon and cytokine-induced antiviral responses are functions of the body's innate immune system. Viruses are only capable of reproduction by attaching themselves to host cells. They reprogram the host cells to produce additional viruses until the disintegration process cell lysis occurs and the cells burst and die. Most animal viruses penetrate host cells intact, entering by invagination of the host lipoprotein cell membranes, in which the viral particle becomes located in a pit coated by the protein clathrin that supports vesicle formation. The resulting closed vesicle fuses with large vesicular endosomes and lysosomes in the process of endocytosis in the cytoplasm. The vesicle is fused with cytoplasmic endosomes and subsequently with lysosomes, which are cell organelles containing enzymes in the membrane-enclosed vesicle. The membrane of the enveloped virus fuses with the endosomal membrane, resulting in eventual viral nucleic acid release into the host cell cytoplasm in an acidic environment involving viral glycoprotein. In non-enveloped viruses, the protein capsid is degraded and the naked viral nucleic acid is released into the host cell cytoplasm during the infection process. Proceed to part 3 of 5.